Thank you, Jim. We're now going to move on to how we brought our industry partners into this project. We're going to talk about skill standards and cell and gene therapy course content. So I'm going to pass it along to Barbara now.
Thank you, Maggie. So if we could go to the next slide. So Russ kind of gave us a rundown of the research we did looking at job postings to understand, is there growth in the industry, and then what are the main skills? But we really knew that we needed to go out and talk to subject matter experts who are in the field in cell and gene therapy to really dive deeper into what skills are needed by our students to make sure that as we develop our curriculum, we are well aligned with industry needs. So we hosted regional subject matter expert listening sessions, and you see the 6 stars there to indicate the 6 regions where we hosted our listening sessions. And we invited local companies in each region that are in the cell and gene therapy space to come meet with us for one day. We had one in person pre-COVID, and then after that, I think we were all Zoom, where we could talk to them in depth about the process of cell and gene therapy, and then really dive deeper into what skills are required for these entry-level technicians. So in the end, if we can go to the next slide, we had 36 subject matter experts across the 6 listening sessions. And you can see here really powerfully all of the companies that had a voice in our discussion, right? So all of these companies at each of the 6 regional listening sessions were able to bring to us their unique perspective based on the processes that are being conducted in their local facilities near each of these regions. So we had, 27 companies that all participated and 3 educational institutions. So for example, here at Juncosa in our listening session, we had representatives from UC San Diego talking to us about their cell therapy lab. Maggie, can we go to the next slide? So our starting point for our discussion were the biomanufacturing skills standards that had been developed by MBC2 back in 2007 and validated in 2013, where we had subject matter experts from what we call now traditional biomanufacturing, which would be the production of protein therapeutics by mostly CHO cells, where, at that time, that was the focus of our emerging biomanufacturing training. So those skill standards were the basis of our conversation, and we would walk through kind of each of the critical work functions and key activities, asking our subject matter experts, okay, does this apply in the cell and gene therapy space, and how is it different from a traditional biomanufacturing work setting? So we focused in on the manufacturing processes, both upstream and downstream. We looked at quality control analytics on the biochemistry and the microbiology side. And then we also looked at issues around compliance and safety to really dig deeper into, what aspects are still pretty much the same when we compare cell and gene therapy to biologics manufacturing, and then where are there key differences that we should have in mind as we train our students. So I'll hand it back now to Maggie and Jim, and they're going to talk to us a little bit about what those differences that we found.
Thank you, Barbara. So that was really fascinating. I enjoyed those sessions so much, and I told the subject matter experts, you're giving us gold. Most of them had come out of what we're now calling traditional biomanufacturing, and they made the transition. And a lot of what we focused on is, what is the difference? What have you noticed as the difference in your career now that you've made that jump? And again, here's Lizbeth, who made that transition going from production of protein to the production of a virus. And so let me share with you some of the things they thought. That it's more complicated, that the product is either a cell or a virus. Those are orders of magnitude more complicated than a protein, which of course is orders of magnitude more complicated than a small molecule drug. The production scale is typically smaller than any of them we're used to. It's closer to a laboratory scale, or maybe for allogeneic, a pilot scale, but it doesn't have the 12,000 or 25,000 liter bioreactors that you might be using to produce antibodies. And then you're going to have a virus somewhere in the process. The virus is the product for gene therapy. The virus is utilized in cell therapy. Next slide, please. I think next slide, I think you went, yeah, next slide, yes. Good, and they made the point that cell and gene therapy is where monoclonal antibody production was 20 years ago. That is, everyone is still figuring it out, especially in the analytics. The techniques are still being developed, new analytical techniques, they're evolving and improving. If you ask the question, what is the critical quality attribute, and that concept comes out of quality by design, what is the critical quality attribute of something as complicated as a cell? That isn't a simple question to answer. And so what does it mean that this cell is ready to go into a patient? That is really still being worked out. Now, I'd say it's in its infancy, but you have 5 different products that are fully approved by the FDA and that are going into patients. And so they worked it out enough to satisfy the FDA and to satisfy themselves that they can have the quality control assurance that these are safe and going into patients. Safe and effective, and they do work. The FDA is, I've been observing them, and Peter Marks, the head of CBER, is, because everything's Zoom, he's attending pretty much every conference that I'm attending, and the FDA is learning along with the industry. They have hired a whole bunch of new postdocs in order to go into this industry, and you feel that they are working as a partner rather than a regulator. That's just a feeling that I get at every meeting, and that regulatory environment is still developing. Next slide, please. Right, and again, this is more complicated. The product is that much more complicated. Exclusively single-use equipment. Time is essential. So again, especially for CAR T therapies, you are on the clock. The patient is waiting. You don't have the time. And criticality, there is a special urgency. You do not get a second chance. And we had subject matter experts that said, this is not for everyone, that you recognize the importance. It isn't like a batch that if you make a mistake, you can make a new batch. You get one shot at this for the patient. And so it takes nerves of steel. It takes someone confident, maybe even a little arrogant, that I can do this and I can do it every time for every patient. And they said of that 2-week period, there is a specific 5-hour period that is just ultra high tension. This is where the major risks occur. If you look at the risk management along the entire length of the unit operations, this is where it is. And the technician has to be especially confident, especially attentive, cannot make a mistake in this particular 5-hour period. And so the stakes are high. Next slide, please. Yes, in autologous CAR T therapy, the source material is limited and precious. So in quality control, really the smallest volume you can pull, the better, because every part of your product that you are taking away to test for quality control is a part that is not going into the patient, and you don't have that much to start with. And so every microliter of the source material is precious, and therefore the analytical techniques that you're using often you are going to devise them to use really small volumes. There are some new pieces of equipment. For example, flow cytometry is important. There are some golden oldies, chromatography, but in the case of adeno-associated virus, the chromatography is not only ion exchange, but the chromatography also is affinity chromatography with a camelid antibody that binds to the adeno-associated virus capsid protein, grabs onto it, you wash through the impurities and then release the adeno-associated virus. So it is the same, but different. And there are new innovations, especially in the analytical techniques that we'll talk about the next time that we see you in January. As I mentioned, it's scale out rather than scale up. And to produce the viruses, you do have a master cell bank and a working cell bank. It's the same to do that, but again, you have these unique transfection steps that you don't have in traditional biomanufacturing. Traditional biomanufacturing, your research department will transfect a bunch of cells, choose the best one, the one that produces the most protein and the one that divides the best, and make a master cell bank out of that and use that for the entire life of the product. That is, someone else genetically engineered the cells. In this case, genetic engineering of the cells is an integral part of every manufacturing process, with the exception perhaps of allogeneic CAR T therapy production. And so that makes a difference. You have these molecular biology steps in the middle of your manufacturing process. I think of biomanufacturing more like chemical engineering than molecular biology. In this case, you have the molecular biology built in to the manufacturing steps. Next slide, please. Good. Downstream is different. Again, for CAR T therapy, you cannot terminally sterilize. It has to maintain sterility. In traditional biomanufacturing, you're going to put it through a 0.34 or 0.22 micron filter at the end, and you're going to sterilize after formulation. Here, you cannot sterilize. It has to remain sterile the entire way. Cells are broken open by chemical means rather than physical means. That is, you add detergents, Triton X or Tween, and you don't put them through a French press. And so that is different analytical techniques. qPCR and drop digital PCR are important in gene therapy and measuring the virus titer. Different data, different analysis, and especially different is rapid sterility testing rather than the testing that is dictated by the US Pharmacopoeia, where you would take a sample at the end and inoculate broth, or inoculate microbial broth, several different types of microbial broth, and incubate it for 2 weeks. You don't have 2 weeks. You're going to do that, but that incubation is going to occur while your product is in the air on the way to the patient. And Barbara says, yes, HIPAA compliance. And so if you are producing CAR T from a patient, there are HIPAA considerations, especially confidentiality, and that you don't have in traditional biomanufacturing. And Barbara said attention to detail, confidentiality, dependability, and that you certainly will require of anyone who's involved in the manufacturing of these. And I think that's the last slide. I'll make this point. Aseptic technique, aseptic technique, aseptic technique. Your students have to be able to transfer materials from here to there without contaminating it. If they can work if they're a master of the biosafety cabinet, they will never be unemployed. All of these companies are trying to develop closed systems where you get the patient cells in a bag and then you sterile weld it, and then you sterile weld again, and you sterile weld again, and you never have to open it up, even in a biosafety cabinet, but still a lot of the processes are open. A good manufacturing practice, again, good manufacturing practice, documentation, quality by design, quality assurance, facilities, personnel training, change control, data integrity, operating your unit operations in a state of control. Every process and every piece of equipment has to be validated that is proven to carry out what you claim that it can carry out, and knowledge of process development and the generation of a design space. The regulations are evolving and keeping on track of those, but good manufacturing practice, good manufacturing practice, good manufacturing practice. Good. Cell and virus banking when possible, robust in-process testing and final product testing by quality control. Again, trying to keep the volumes down. The regulatory requirements are interesting. So if you think of what the future of CAR T could be, why can't every hospital manipulate their patient cells on site? That is, to have a distributed model rather than collecting the cells, sending it to Seattle and Bristol Myers Squibb or to El Segundo and Kite. Why that centralization? And a lot of that centralization comes down to the regulatory requirements, operating under good manufacturing practice. Every hospital isn't really prepared for a GMP facility, and the expense and the effort and the expertise it would take to convert that to GMP. Some of the big gigantic universities can do that, but not every community hospital. And so the regulatory requirements are driving, I think, this centralized production. Next slide.
You think
and let me hand it off to Maggie.
Okay.
Thanks, Maggie.
Thank you, Jim. So just to conclude, and that was great, Jim, we got great information from our subject matter experts, from our industry partners, and we're going to publish a skills standards report with this information, you know, what's different, what's the same as traditional biomanufacturing. So we're working on that now, and that should be published in January. In addition to having the SMEs review the skill standards, we actually gave them some open-ended questions. We really wanted to learn from them, what should we be teaching? So if we're developing a course on gene therapy or a course on cell therapy, what's the foundational knowledge? What are the skills that are required? And even what are the non-technical skills? So we got, again, a wealth of information from these open-ended questions that we gave to these 36 SMEs, and we've compiled some of them here in the following slides. Some of them are not surprising, since you've been hearing a lot about the production of cell and gene therapy throughout today. And some of them we're already teaching our biotechnology programs and courses. So if you were to develop a course in gene therapy, some of the foundational knowledge they would like us to incorporate is cell biology, genetics, maybe increase the immunology that we teach now and introduce more virology. So maybe some of our programs where we don't go into much detail in virology, we focus a lot on microbiology. So increase the virology, the details, and how viruses are produced, what genes are necessary for viruses to replicate, to build a virus. And then basic concepts of biotechnology and molecular biology. Again, this is not surprising. In order to produce a viral particle, gene expression, plasmid construction, plasmid designs, transfection and transduction processes, gene sequencing, qPCR, quantitative PCR, gene editing methods, and knowledge of viral vectors. So these are some of the basic concepts and the foundational knowledge that we should be thinking about as we design these courses. They also wanted us to focus on the landscape of gene therapy, the types of gene therapy, types of viral vectors that are used. You know, we've heard today that for the most part, it's adeno-associated viruses that are used to make these products. How are they designed? What's the gene therapy approach? Is it in vitro, ex vivo, in vivo, ex vivo? The fundamentals of the process of gene therapy, and you've heard a lot about that today from Jim, and how that differs from traditional biomanufacturing. We covered the similarities, validation, for example, aseptic processing, but what are the differences, and focus on them. Focus on process understanding, and particularly what's the impact to the patient. So many of these companies and pharmaceutical companies in general really want their employees to focus on, this is for the patient, this is to help the patient, and it's very critical in autologous gene cell therapy, but it's also critical for producing gene therapies as well. Process understanding, how it impacts the product, how it impacts the technician themselves, the individual making the virus, how the process impacts, again, the product and also the environment. Regulatory environment, the FDA, what regulations should they be following? CGMP. In addition to that, what other regulations pertain just to gene therapy? And then introduce case studies, talk about patients. And this came up time and again when we talked to our SMEs. They want us to, like Jim did earlier, show patient stories, let them understand that this is curing a child or an individual, a patient that might not survive otherwise. So introduce case studies. And then for the manufacturing process, discuss cell culture. We talked about aseptic technique a lot, adherent versus suspension cells, the scale-up process for gene therapy, how it differs from traditional biomanufacturing. Main point there is that it is entirely single use, is smaller scale than traditional biomanufacturing, or the use of aseptic connectors, tube welders, for example, and the understanding of chromatography and chromatography skids. The knowledge of the properties of the viral product, again, looking at process impacts. What should you be thinking about so you don't damage and you get a viable, effective viral product? Just like we learned about, you know, characteristics of proteins that can be altered through the process of traditional biomanufacturing. Discuss infectivity and titer determination. So you're generating a viral product. Is it viable? Can it infect other cells? And there's ways for us, and we're developing some curriculum on that now, looking at transducing CHO cells and other cell lines, and then evaluating the transduction efficiency and effectiveness. Learn about titer determination, again, chromatography for purifying the viral product, tangential flow filtration, ultrafiltration, diafiltration, and then, as we've already learn from traditional biomanufacturing, batch records, the importance of batch records. Electronic batch records is mostly where companies have gone now. But even having, you know, hard copy batch records as we teach, just understanding, following a batch record to the T, also digital lab notebooks. CGMP, again, is similar to traditional biomanufacturing, very important. Environmental health and safety, there's some other issues we have to worry about, as Jim mentioned, or we have to teach, because we are purifying a viral product that can infect human cells. And that goes along with biohazard waste disposal, and that came up quite a bit as well. So what lab skills should we be covering in this gene therapy course? This is one of my students here performing an electroporation experiment in a biological safety cabinet. Again, aseptic technique is key. Cell culture knowledge, how you scale up cell culture in single-use instrumentation. DNA extraction, plasmid construction, plasmid purification. Much of this we already teach in our courses. Transfection, transduction, gene editing, gene sequencing, plasmid sequencing, when you introduce you make your plasmid construct with your transgene. Techniques like Gibson assembly. How do you construct a plasmid? What's the most efficient way of introducing a transgene into an expression vector? Single use, as we mentioned already, and purification. They particularly like to, some of us are using the ACTA systems with Unicorn software. That's something that they use in industry. Ion exchange, affinity chromatography, and ISO is going to talk about that in just a second. And then analytical techniques, digital droplet PCR, a form of quantitative PCR that's is mostly used now in the gene and cell therapy industry to quantify expression of transgenes. SCS Page for analyzing purification, ELISA, HPLC. Again, these are techniques that we already teach, but developing protocols, as we are doing now, that allow us to use these techniques in the realm of gene therapy production and analytics. And then, of course, potency assays, which are very specific to the particular gene therapy. So this is the information that we got when we asked our subject matter experts and industry partners what we should be teaching in a gene therapy course. And some of them most of you can already check off as something you're already teaching your students, but we can also adapt those techniques to make them relevant to the gene therapy space. We also asked them what topics should be covered in a cell therapy course, and the foundational knowledge here was more immunology, because many of the products, as we've learned today, that are in the pipeline deal with immunological cells like T cells, natural killer cells, and also stem cells as well that are being differentiated into some of these cell types. Cell biology, immunology, and virology. So there's some overlap there with the gene therapy course. Then discuss the landscape of cell therapy. Jim nicely explained allogeneic cell therapy and how it compares to autologous cell therapy. The different types of cell therapy. Today we've focused a lot on CAR T, but of course there's stem cell therapies, there are cell therapies that have been in development or available for a number of years already. And then biotechnology and molecular biology, basic concepts, again, to talk about introducing transgenes into these cells by electroporation or through lentiviruses. And then the fundamentals of biomanufacturing. So this is similarities to traditional biomanufacturing, and then the key differences that Jim has really emphasized as he went through what we learned from the skill standards review. Scale is different. Criticality is very different in that it's for autologous, especially, it is that patient's one chance of treatment and survival, perhaps. And then the variability of the starting material no longer are, unlike traditional biomanufacturing, you're not going to the cell bank and getting your vial of cells and growing them up. And if something goes awry in the process, you can then terminate that batch and go back and get a new vial. So that's certainly not the case with autologous cell therapy. Introducing the methods and equipment used for apheresis, so isolating the T cells to be used, using cell sorters, using Ficol-based gradient centrifugation post-apheresis to isolate that T cell population, activation of T cells in vitro, and then again the regulatory environment. And for the manufacturing process, again, I just covered this already, but really that understanding of the small scale, the preciousness of the cells that you're working with and the timeline that you have to work with them. Data analysis and process trending is something that came up often because each batch is slightly different, partly because each patient that the cells are coming from is at maybe a different stage of treatment of their disease. So looking at data as you're going through the process and analyzing that data, making decisions, changes might have to be made in a production. One production is different from different from another production because of this variability of the patient sample. So data analysis is very important, and process trending. Recognizing the direct impact on the patient, again, single-use systems, and Jim brought up chain of custody, chain of identity. So this differs from traditional biomanufacturing because of the autologous nature of the CAR-T therapy that's in production right now. So there can be no mix-up. So chain of custody, chain of identity is critically important. We talked about the variability of the cells we start with autologous, and then operation of cell culture equipment, upstream processing equipment, and then learning about cell washing the cells before they're reintroduced back into the patient, for example, and the type of equipment that's used for that. So even if we can't purchase all of the equipment that would be used in a CAR-T production facility, we can teach the students about this equipment so they'll know what to expect and what that equipment does in the process. So what lab skills should we be teaching in a cell therapy course? The lab skills we can teach with non-highly specified or specific equipment are things that we do already. Again, aseptic technique and cell culture, very important. Cell expansion, growing the cells in shake flasks or tea flasks, expanding them to single-use cell culture bags on a rocking platform, still at small scale, maybe a liter of cells, for example. Learning about tube welding and how you aseptically transfer cells from one vessel to another. Again, it's much smaller scale, so our lab scale work is absolutely fine here. Gene delivery systems, electroporation lentivirus. Electroporation is something that came up because some companies are considering this method of introducing transgenes to modify T cells. CRISPR, of course, is important for them to know because that is becoming a big part of this process. Then analytical techniques, flow cytometry is very important in this space, and we are developing some standard operating procedures and some experimental processes to introduce your students to flow cytometry. qPCR, ELISA, and then Jim mentioned also sterility testing. So because of the time constraints of autologous cell therapy, sterility tests are very quick. Results are known in hours and not days. And this has led to the development of new technology and new equipment so that sterility and highly validated sterility tests can be done quickly so these cells can get back into patients in a timely manner, and of course mycoplasma testing. So those are some of the lab skills that we'll be developing curriculum and sharing that with you so you can introduce some of these skills into your courses or programs. We also asked our industry partners about important non-technical skills, and these are skills, some of these are skills that have come up time and again when we talk to our industry partners and our hiring managers. But a very important one was attention to detail, and that's why it's the first bullet here. So many of our partners indicated that this is key, and from what you've heard today, you can understand why. Attention to detail, following instructions, following batch records and protocols. Another important one, because of the criticality of some of these production processes, is honesty. And if something goes wrong, you be honest and you communicate that to your supervisor. And to be conscientious, so to be, following details, following instructions, be conscientious, be invested, and have a patient-centered mindset. That's something that came up time and again as well. Interpersonal skills, good communication skills, good teamwork skills, being able to collaborate with others, always important. Ability to work in a fast-paced environment and cope well with change. Again, working on a timeline for autologous cell therapy, when things may change, and to be able to adapt and change when necessary, a certain process. Ability to follow written procedures and documentation, good documentation. So being able to document your results and be able to document what you're doing as you follow a batch record, critically important. Learning agility, this is something that came up, being able to be willing to have self-development as far as continuing to learn on the job, learning your skills as you evolve in that position. And then an efficiency mindset. This really comes down to good time management, but this was mentioned by a few of our industry partners, being able to look ahead and prep for the next step. When you're waiting to take a sample, for example, obtain the sample collection labels, gather your materials, get ready for that next step, make very good use of your time. And then lastly, how to maintain consistency each and every day when performing an assay or performing a process. Again, very important, such that each batch will be of the highest quality and no variation. So taking this into consideration, what we learn from our subject matter experts, we're currently developing some curriculum, and you'll hear about our adeno-associated virus production in HEC293 cells today. We're also developing curriculum using SF9 cells and developing a recombinant AAV that's expressed in GFP in insect cells, SF9 cells, and Thermo Fisher have basically a kit you can buy. We can buy the cells and the transfection reagents, and you just have to add the recombinant baculovirus. That's not commercially available at the moment. So Jim and I are reaching out to labs that have developed recombinant baculovirus for GFP to obtain that, so we can develop that curriculum and infect SF9 cells. Of course, the benefit of SF9 cells is that they're biological safety level 1. So we'd like to be developing that curriculum in parallel with ISO's HEC293 curriculum. We're developing protocols and SOPs for cell transduction and analysis through flow cytometry, and also through simple Western blot to show expression of transgenes. And we're developing analytical assays, qPCR, ELISA, HPLC, and STS-PAGE for analyzing the AAV viruses after being produced in our systems. In addition, we are developing SOPs for single-use bioreactor setup, operation, and aseptic connections. So that's what we have in the pipeline right now for gene therapy, and we're going to share all that curriculum on the NBC2 website. Cell therapy, aseptic technique, so activities to improve the skill level of your students after they've been taught, the aseptic technique and cell culture. Gene delivery techniques, electroporation, again, lentivirus, and flow cytometry analysis of cell therapies. So again, this is curriculum we have in development now. And you can find that you will be able to find that curriculum on the NBC2 website at biomanufacturing.org. And that's where we have right now our labor market report that Russ described earlier. And also that's where the skill standards report will be as well. And I will also have that on the Innovate Bio website as well. So that's where you can find much of what we talked about today. Some of it's still in development, some of it's ready to be shared. So I'm going to pause there to see if there's any questions on our skill standards development and basically what we learned from our subject matter experts.
There were a couple of questions, Maggie. One question was, should we be talking to our administration about a BSL-2 lab, or would insect cells be sufficient for industry?
The reason we're working with insect cells, because we could all, the partners in the grant could all just adopt the HEC 293 production, because as you hear from Izzo, he has it pretty well worked out already, but Jim and I wanted to give people a biological safety level 1 option. Depending on your college, some are resistant for labs to be using biological safety level 2 cells or organisms. So, but getting back to industry, when we talk to our industry partners, some say it's good to learn both, and others say we're going to stick with HEC 293 cells. For teaching purposes, we feel that using insect cells is absolutely fine. They're still getting the viral particle production. They're understanding what genes have to be introduced into the cells in order to produce the recombinant viruses, and we can then collect the viruses and use them to transduce and infect biological safety 1 cell culture cells like CHO cells or mouse fibroblast cells. So we can do the whole process in biological safety level 1 and teach the students everything they need to know.
But to me, it seems to me that it depends on what your local industry is doing. So if your local industry is using HEC 293, then it may give your students another leg up in order to get hired there. Around us, it's mainly insect cells. So.
And then the other question is plasmid construction an entry-level skill for manufacturing?
That's an interesting question. And the answer is yes and no, right? So plasmids in this case, they call them a starting material rather than a raw material. So here you have plasmids being produced usually by a different company and then being purchased by the cell or gene therapy company. And there is a major shortage of workers that can produce plasmids at a large scale. And remember, in the Moderna and in the Pfizer COVID vaccines, an integral part of that process is starting with plasmid that contains the gene for spike protein. But I've been thinking about this a lot, that 10 years ago, I was saying that in biomanufacturing, the product is protein, and protein is the cash and DNA is the flash. And then we are making this transition that, well, the product is protein. And I should say, remember, although we're getting excited about cell and gene therapy, monoclonal antibody production is going to dominate for the next decade. And that is going to be the dominant. So we can't forget about training students for what we were doing before, because we're excited about this, because it's going to remain to be dominant. But now the next step is going to be, I think, training students to produce plasmids as a product. And again, as a product that will become a starting material for what we're doing here. And basically what they do is a plasmid prep. It's an alkaline lysis of E. coli plasmid prep, but at a big scale. And so I'm thinking we should have some demonstration sites of scaling that up, especially with our buddies in St. Louis, where Pfizer produces their DNA. So that's a long-winded way to say, yes, plasmid production is very important and emerging, and there is a shortage of facilities that can do that. So when you do a plasmid prep, remind your students that this is not only now for producing plasmids for research, but if you were to scale this up to hundreds of liters, you would be producing the starting material for a COVID vaccine.
Thanks, Jim. So perhaps we can answer any other questions in the chat, and happy to do so. So we can move on to